Today we're going to have a look at enzymes, more specifically how they work. So enzymes are globular proteins and they're roughly spherical with their hydrophobic groups pointing inwards and their hydrophilic groups pointing outwards. This means that the hydrophilic groups will be able to bond with the water in the surrounding environment, making them soluble in a solution. This is very beneficial as it helps to transport enzymes throughout the bodies of humans and animals. Now, enzymes have several different components to it, but one of the most important components is the active site. The active site is very specific and has a very particular shape. Our enzymes are known as biological catalysts. They're biological as they're made from a biological molecule called protein, and they're catalysts as they are able to speed up a reaction. Now, enzymes catalyzes virtually every single metabolic reaction within living organisms, including humans. Enzymes can be either intracellular or extracellular. Now, intracellular enzymes are produced, manufactured, and function within the cell itself. Now, one example of this type of enzyme is DNA polymerase. Now, DNA polymerase is responsible for the DNA synthesis within our cells during cell replication. Another example is ATP synthase, which is an enzyme responsible for the synthesis of ATP, that is an energy carrying molecule used throughout our body. Extracellular enzymes are produced and manufactured inside of the cell, but they're secreted and they function in the external environment. Now, one example of this is amylase. Amylase is secreted outside of the cell, usually the salivary glands, which is responsible for the breakdown of starch into amylose in the mouth. Enzymes can also be classified as either anabolic or catabolic. Now, anabolic enzymes synthesize larger, more complex molecules from smaller ones, typically requiring energy input in the form of ATP. One example of this is glycogen synthase, which is responsible for the synthesis of glycogen from glucose in the liver. Another example is RNA polymerase, which is responsible for the synthesis of mRNA from DNA in protein synthesis. Now, catabolic enzymes break down these large molecules into smaller ones. These reactions usually release energy in the process. This energy can then be stored as ATP. One example of this is lipase, which breaks down lipids into fatty acids and glycerol. And another example is almost all the enzymes responsible for cellular respiration, the main metabolic pathway for producing ATP. Before we can understand the mode of action of enzymes, we need to understand reactions and how they occur without catalysts. Now, atoms move. In a gas state and in a liquid state, they move pretty fast, but in a solid state, they vibrate. Now, for a reaction to occur, two particles need to collide at a specific orientation for the bonds of each particle to break and to reform new bonds. So it needs a specific and sufficient amount of energy to break these bonds. This energy is called activation energy. We can define activation energy by looking at energy diagrams. So first, let's look at the energy diagram of a catabolic reaction. Now remember, catabolic reactions are the breakdown of molecules and they release energy. So we can graph a catabolic reaction as the amount of energy over the progression of the reaction. The first bit would be the energy of the substrate, and this will be quite a high amount of energy. The second energy will be the energy of the products, and this will be a lower amount of energy as the reaction releases energy, so the energy will decrease. Now, we would expect the graph to just decrease from the substrate to the reactants, but this does not occur. First, the energy increases and then decreases. This increase of energy required is the activation energy that is required. We then also have anabolic reactions. Now remember, anabolic reaction is the build-up of molecules, and this requires energy. Now we can also graph this as the amount of energy over the progression of the reaction. Now the substrate energy in this example will be less and the product's energy will be more. This is because it needs to gain energy for the reaction to occur. Instead of just increasing directly to the product's amount of energy, the reaction will require even more energy than the product and then decrease slightly up to the energy level of the products. Now, the energy required by the reaction to occur, or the activation energy, can be supplied by heat. So if we increase the temperature, we know that we will also increase the kinetic energy. Kinetic energy is increased, the particles will move quicker. This will cause more collisions to occur and more reactions to occur. Our bodies kind of already do this. Our body temperature is at 37 degrees Celsius, which is much higher than the external environment. But this is not enough for the reactions in our body to occur. So our body also uses enzymes. Now, enzymes are able to increase the rate of the reaction by decreasing the activation energy needed for the reaction to occur. Here we have our enzyme. Our enzyme has a substrate binding site, also known as the active site, that would have a shape complementary to the substrate. So our substrate will move into this binding site and fit into it perfectly, called the lock and key model. Now, the lock and key model says that only substrates with a specific shape can fit into the active site. It also says the shape of the active site cannot change and that the enzyme will remain unchanged during the reaction. But there's some limitations. We know that enzymes can adjust to better interact with their substrate, and for this to be able to occur, our enzyme must be able to adjust its shape. We also know that certain enzymes can interact with multiple different substrates as well, which means that our active site cannot be as rigid as previously thought. And we also know that our enzyme is able to bind to other molecules that have a slightly different shape to the substrate that's needed. So to account for this, we use the induced fit model. This model says that the shape of the active site can change slightly. So we call this change a conformational change, and this change occurs while the substrate is binding to the enzyme active site so that there's a better interaction between the substrate and the enzyme active site. So this slight different conformational change ensures the ideal binding arrangement of the substrate to the active site. Here we have pepsin as an example. Now, pepsin is an enzyme that's responsible for the breakdown of protein into amino acids in the stomach. Here we can see in the 4 examples of pepsin given, in each one the active site is slightly different in shape. The substrate is able to bind to the active site, and the enzyme will undergo a conformational change to fit the enzyme perfectly to optimize binding between the enzyme and the substrate. The enzyme is then able to orientate the reactants and bring them into close proximity. This reduces the need for multiple and random collisions. The enzyme is also able to distort and weaken the bonds, which lowers the energy needed for the reaction to occur. In other words, it lowers the activation energy. Our substrate will then leave the enzyme as products. In this case, amino acids will be released.